Our next speaker is Dr. Blayne Amir Saeed. Dr. Saeed is a member of the Division of General and Thoracic Surgery at the Hospital for Sick Children. He specializes in malignant and benign diseases in the liver and biliary tract. Dr. Saeed is dually certified in hepatopancreatobiliary surgery and abdominal organ transplantation with particular interest in minimally invasive liver surgery, liver donor liver transplantation, and complex liver surgery involving vascular and biliary reconstruction. He is an associate scientist track with a background in cellular immunology. His presentation is entitled Ninjurin-1 Contributes to Lytic Cell Death in Hepatic Ischemia-Reperfusion Injury. It will be a 10-minute presentation with questions afterwards, or 20-minute presentation with questions afterwards.
Glad you said 20 minutes. Otherwise, I was going to have to go really quickly. Nice to meet everybody. Thanks for having me here. This is actually my first CDTRP meeting. I joined faculty at University of Toronto 3 years ago. So soon to follow by COVID and the pandemic. So it's nice to be at meetings in person in general and to put some faces to names that I've seen along the way here. So in terms of, I'm going to talk to you a little bit about some of the work that we're doing in my lab, which is in the program in cell biology in the Sick Kids Research Institute, and in particular talking about ischemia-reperfusion injury. So, you know, the problem that we deal with in liver transplantation and in solid organ transplantation in general is that when we take organs out of people's bodies and put them to other people's bodies, cells die, just to put it bluntly. And this is still one of the biggest hurdles that we have to overcome in transplant. It's the underlying premise behind all of the talks that you're hearing in terms of optimizing this surgical procedure. In liver transplantation, it really has a profound, this ischemia-reperfusion injury has a profound impact on graft function. When we use organs that are outside of our kind of typical perfect organs, it has a more pronounced effect. And certainly for patients who are sicker when we transplant them, it has a very potentially devastating effect for those high-risk recipients. And why this matters so much in liver transplantation is, in essence, we're able, this is just looking at some SRTR data from the US from 2019, but in essence, at any given time during any given year, we're able to transplant only about a third of the patients on the liver transplant wait list. And unlike other forms of organ failure, where you have potential hemodialysis or you have ventricular assist devices, in liver transplantation, the outcome is in essence binary. Either you're going to get transplanted or you're going to die. We remove very few people from the wait list who improve and don't need a liver transplant anymore. That happens quite rarely. Sometimes they have cancer and it progresses, so they're no longer a transplant candidate, and then you're going to have a poor outcome, or they're too sick. So this continues to be a big problem. This data, just looking at that data in the US, you know, despite the fact that we try to recover and use more organs, we end up discarding in the US about 10% of that. So people are trying to push the envelope, but the reality is we hit limitations. And the reason that this matters is if we're trying to push the envelope, use organs that are a bit more marginal, you can end up in a very bad situation. And so when you look at patients who have moderate or severe ischemia-reperfusion injuries, there's data from UCLA's program, you end up with significantly decreased 1 month, 6 month, and 12-month survival of those organs and those recipients. So you can try to push the envelope by using more marginal organs, but you oftentimes, or to be more accurate, the recipients oftentimes end up paying a price for that. And that can result in significant multi-system organ dysfunction and potentially death. So what does this look like? So basically, if we have a perfect organ, in general, we don't worry so much about cold ischemic time. We don't worry about how sick a recipient is. But anytime we start to deviate from that, so when we have organs that are coming that are a bit more marginal from older donors, from livers that we know are steatotic, but that do work in those donors, from DCD donors, and coming from far distances, the more risk factors that we accumulate, the more likely it is that we are to have an ischemia-reperfusion injury. So what happens? So just from a basic cellular level, when you interrupt blood flow and blood supply to that organ, cells die. And with that dying off of those cells, you have release of intracellular products. Those intracellular products are very brisk pro-inflammatory mediators. And so with that, LCD is lytic cell death. So with that first round of lytic cell death and release of these DAMPs, these pro-inflammatory mediators that are contained inside the cell, when those are released, when we reperfuse the organ in the recipient, a couple of things happen. The first thing is it can make the recipients very, very unstable and quite sick. This happens immediately intraoperatively. It can be a challenge to deal with. We've lost patients certainly on the table for that reason alone, or I had patients who end up in the ICU critically ill afterwards. The second thing is that you already have a very stressed organ and stressed cellular system. And so it causes a second round of cell death in that organ. And those it becomes this kind of crescendoing cycle of cell death, creating more inflammation, more cellular stress, and more cell death. Long term, with those repeated rounds of lytic cell death and release of the DAMPs, you end up with prolonged graft dysfunction. You can end up with early fibrosis. You can end up with graft failure, requiring retransplantation. So it's really quite a problem that we deal with. And the reason that matters is we're always trying to expand our donor pool. We're trying to use more of the offers that come in. One thing that's so frustrating as a transplant surgeon and a transplant professional, as we all deal with, is you want to use the offers that you get. And oftentimes you're really faced with difficult decisions about, can I use this organ safely in this recipient or in any recipient? So do we have potential targets to help us with this? We have a lot of technical solutions, some of which we've heard about today. And in particular, in liver transplantation, we are doing ex vivo perfusion. Right now, a lot of that's telling us whether or not the organ is good enough or not good enough to use, but it's not necessarily making the organ better. And so we have a lot of potential targets and a lot of room for therapeutics to improve organs when they are on our ex vivo systems. And something that my lab is interested in is lytic cell death is actually a very active process. And are there ways of inhibiting that active process in order to minimize the pro-inflammatory DAMP release and also to preserve cellular integrity and liver parenchyma and therefore improve liver function? It's a little bit of a busy slide here, but this is basically this is a relatively recent update. So that wheel on the left, so basically before we used to think of apoptosis or necrosis. The wheel on the left shows all the different forms of regulated cell death, that's RCD, that we now know. So there's a whole variety of actually very distinct types of cell death. Some of them share common molecular machinery, but have different stimuli or have different intracellular factors that regulate them. And what oftentimes happens is, this happening all the time in the body system, is that you have perturbations of the environment. That's that green bars on the right. When you have perturbations that the cellular environment can't deal with, you end up with lytic cell death. And that's an active regulated form of cell death. And then that stimulates more inflammation and more cell death. And so those points at which we have lytic cell death are actually target points for a treatment of organs, particularly in the context of transplant, that if we can abrogate some of those factors, that we can improve graft function. This is a paper that came out in 2021 in Nature from the Dixit group at Genentech. And basically, they identified this molecule called NINJ1. So basically, this is just looking at bone marrow-derived macrophages from mice. It's the only Nature paper I've ever seen that only uses one cell type, just in vitro assays, just bone marrow-derived macrophages in mice. But they saw something really profound, is that when they had these knockout animals that do not express the NINJ1 molecule, and that's in the red bars, when they induce various forms of cell death, an inflammatory form of cell death called pyroptosis, necrosis, apoptosis. Across forms of cell death, multiple forms of cell death, these NINJ1 knockout cells were protected against cell lysis. The left-hand bar, the axis there is just basically LDH release, which is a marker of cell lysis. And so these NINJ1 knockout cells are protected from lytic cell death across various forms of active lytic cell death. So a really interesting finding. And so this is the model that they came up with, which involves NINJ1 being a large drill, which I really like. There's no evidence that's actually a large drill, but it's the way that they made this schematic. But this is basically showing downstream effects in one form of lytic cell death, which is pyroptosis. So when you have, like I mentioned, infections or those sterile insults or the release of intracellular contents, when they're picked up by a cell, they they trigger the formation and the activation of something called an inflammasome, a multi-protein complex, which then serves as a substrate for caspase. These are very pro-inflammatory molecules. They cause a cleavage of this gastromin D molecule, which assembles into this nice pore, and that allows release of pro-inflammatory cytokines like IL-1 beta, IL-18 out into the environment. Even in the absence of that pore, that yellow gastromin D pore, cells don't rupture. However, you get cell swelling, but the cells don't actively rupture. So you can have pro-inflammatory cytokine release, you can have cell swelling, but they don't rupture. The rupture is really dependent on this NINJ1 molecule, which they don't know what it does, but this is how they posit their schematic of how it's potentially contributing. So what we know about NINJ1, this really piqued my lab's interest, some of the folks that I work with. So it's a transmembrane protein. It's actually been known about for a long period of time. It was thought to be important for neuronal signaling. And it is a 16 kilodalton protein. It's in the plasma membrane, has 2 transmembrane domains. And it has actually a very broad tissue expression profile. This didn't come across for some reason in the graphic there, but when we look, this is data from Sonia McParland, who's an investigator at the TGHRI, who has basically done single-cell RNA-seq of pre-transplant liver grafts, so healthy livers. It's very enriched NINJ1 expression in macrophages and hepatocytes from these normal human livers. So it's widely expressed in mice for sure, and in rodents, but also in humans as well. And in particular in the liver, it's highly expressed in macrophages and hepatocytes. So we used in our lab, we got the NINJ1 knockout mice from the Dixit group. And we just use a fairly simple model, which is an ischemia-reperfusion model in vivo, where we clamp the blood supply to 70% of the liver for a period of time, here an hour, and then we have reperfusion for 6 hours. And then we look at indicators of injury. So when we use these NINJ1 knockout mice, we see a couple of things. There's the sham, we just have a sham laparotomy in those animals. But when we look at AST and ALT, those are both liver enzymes, not entirely specific to liver, but are most broadly used molecules where we measure liver injury. We see significantly decreased circulating serum levels of AST and ALT in these animals post-ischemia-reperfusion injury. Similarly, we see decreased levels of LDH in those animals. Again, just a marker of a cell rupture. This is a native page gel, and I'll talk about this, why this is important. But when we look, this is in a wild type liver specimen. When you look at a sham versus an ischemia-reperfusion injured model, what we see is there is a shift towards high molecular weight bands looking at NINJ1. NINJ1 itself in these native page cells seems to exist as monomers or dimers mostly, but when you have an activated injured liver specimen, you see oligomerization of NINJ1. And this is seen by the Dixit group as well, again, just in macrophages, but here we're seeing this in whole liver perfusate. When we look at the necrosis that we see in these animals in the livers, sorry, this didn't come across either so well, but we see a similar pattern. Basically, it correlates with what we're seeing in the serum, that we see decreased levels of necrosis, tissue necrosis in the liver. There seems to be some threshold, like if you get above a certain percent of necrosis, even in knockout animals, you can see tissue damage, but you get a much more patchier, less dense necrosis post-ischemia-reperfusion injury. And this is a pretty profound injury in the wild type animals. If you had this in a whole liver, 60-70% necrosis, you would not see survival. When we look in the serum, we look broadly for serum cytokine and chemokine expression. What we generally see is not a lot when it comes to cytokines in particular, but for chemokines, these are chemoattractant factors that lead for infiltration of the liver or the target organ with immune cells afterwards. We see a fairly specific and reproducible pattern where we see CXCL9, which is otherwise known as MIG, and IP10, as well as eotaxin. The only cytokine that we see that really gets upregulated is IL-6. And in the knockout animals, that's the third bar from the left. We, in comparison to the wild type counterparts, we see significantly decreased levels of those early pro-inflammatory cytokines and chemokines. And interestingly enough, this is a paper from the Penn group, Kim Moltoff's group at Penn, but they see a very similar pattern, particularly with IL-6, IP10, and MIG in patients who have early allograft dysfunction. After liver transplantation, they see similar of those specific cytokines and chemokines. So a similar phenomenon with early liver injury in that context. So what exactly is happening and how does NINJ1 regulate this? Well, we know, interestingly enough, that NINJ1 has two potential distinct functions. So the early data from NINJ1 was basically all about function 1, which is immune cell adhesion and migration. So there's really good data, predominantly in mouse models, but looking at the lung, looking at the central nervous system, that activated NINJ1 aggregation via homotypic or heterotypic binding basically allows for increased infiltration into the target organ, and that there is an inhibitory peptide called NAM, which blocks that. The plasma membrane rupture, which was newly described in that Nature paper in 2021, is a totally new and distinct function. People hadn't heard about that before. The mechanism, like I said, is unclear, although it does require NINJ1 aggregation. And it seems to be mediated by that alpha helix, which is in red there, that I've labeled lysis. It seems to be mediated by that alpha helix, and that's by targeted mutation data from the Dixit group. Interestingly enough, and I'll show you some of this data, it seems to be inhibited by glycine. So glycine is a pretty simple amino acid, but particularly in the transplant community, we've known about its cytoprotective effects for decades. When the Belzer group in Wisconsin was putting together some of their early organ preservation fluid data, they actually used simple amino acids, and glycine was one of them. And for whatever reason, has a very reproducible effect in terms of cytoprotection. The mechanism of action has long been unclear, no clear and good explanations. We've used it in transplant and hepatobiliary surgery intermittently, again, as organ perfusate, organ preservation fluid, and people have even done trials in HPV surgery and clinical trials with good results. But the administration is cumbersome. You have to have it, in essence, constantly circulating, so not very easy to do and requires repeat dosing. It washes out very easily. So when we just look to see if we could differentiate, in essence, if NINJ1 has two distinct functions, this is early data, could we inhibit them and see what the difference is? So if we use this inhibitory peptide, which seems to inhibit the transmigration effect of NINJ1, we don't see much of a difference. This is small groups, N of 4 for each group, but looking at treatment of mice using the same ischemia-reperfusion injury, don't see much difference either between AST, ALT data or percent necrosis. When we use glycine treatment, however, we do see a reproducible difference in AST and ALT levels in terms of circulating. And again, we start to see differences in terms of the percent necrosis in those animals. So suggesting that it is a effect on the rupture effect of NINJ1 rather than the migration effect. This is a paper published by one of my collaborators, Ben Steinberg, who's a clinician scientist, in the anesthesia group at Sick Kids, and this just came out. So basically, if you look at the upper left corner, which is looking at bone marrow derived macrophage from wild type and knockout animals, when we induce cell death, lytic cell death by pyroptosis, so we add LPS plus nigiricin, you get reproducible, very good cell rupture as measured by LDH. When you do it in knockout animals, you see decreased cell rupture, as you would expect, as shown by the Dixit group. And then when we inhibit with glycine, it basically shows with wild type cells, it basically shows you the same phenotype as a knockout, and there's no augmentation of the knockouts. In essence, it doesn't add to the NINJ1 knockout protection. The native page gel on the bottom just shows the same, that basically glycine addition mimics, in essence, NINJ1 inhibition, that you don't see the formation of these heterocomplexes. And in the cells, this is looking by birefringent microscopy in the top right-hand corner. You see with LPS plus nigiricin, you get those clusters, nice distinct punctae where the NINJ1 clusters. When you inhibit with glycine, it becomes much more diffuse. And then looking by, and that's quantified in the bar graph below, and then looking by STED microscopy, you can kind of get a better sense of what those clusters look like. They're fairly large, high molecular weight complexes, which I'm showing here. And so the model that we have is that somehow NINJ1 is clustering in the plasma membrane. The obvious thought process is it's forming a pore or something else that's allowing for plasma membrane rupture, although we don't know yet. The gasdermin D pore that I talked about before is a very large pore. That's only 21 nanometers. And so if you look here how big for this STED microscopy, these are really quite large clusters. And so we have some work going on with some other groups to try to figure out what exactly from a quaternary structure these NINJ1 molecules are doing. Is it a pore? Is it some type of molecular machinery? But very interesting because, again, it's a highly conserved mechanism there. And so the last data that I'll show is important from a liver perspective. So we know that hepatocytes, in which is what are the cell types that are important. So we know hepatocytes and Kupffer cells make up the vast majority of cells in the liver. Kupffer cells are the resident macrophages in the liver. We know that macrophages undergo this pyroptosis, this inflammatory form of cell death that's been widely documented in vitro and in vivo systems. But the role of hepatocytes is very poorly defined. We tend to think of them as almost as bystander cells, and it's not even clear that they contain the machinery to undergo lytic cell death in this context, or that they can undergo inflammatory forms of those lytic cell death. So we've been using basically, it's a little bit cumbersome, but basically isolating primary hepatocytes from mice and then using hypoxic systems. These are true hypoxic systems where everything is contained within the hypoxic chamber, including the media beforehand. And so what we find before we go into the hypoxia stuff is that indeed hepatocytes can actively undergo lytic cell death. That's the bar graph on the right, that when you add in pro-inflammatory, lytic cell death markers like stimulators, that you can induce really nice hepatocyte rupture. In NINJ1 knockout animals, or NINJ1 knockout cells are protected from that. And when you add glycine back into the wild types, you can recapitulate that NINJ1 knockout phenotype. And we see the same thing, although early data, they're a bit harder to isolate with Kupffer cells. And now this is looking in a hypoxic system. So when we basically have an hour of hypoxia plus 4 to 6 hours of reperfusion afterwards, even in the absence of LPS plus nigiricin, we can end up basically inducing lytic cell death in that context, and treating them, either knocking out with NINJ1, which is shown here, or treating them with glycine basically inhibits that function. And so hepatocytes aren't really just plain old bystander cells. They're actually very involved in this process in vitro, we've shown. The last stuff that I'm going to show is basically along the same lines. We know that in macrophages, when you induce this inflammatory cell death, you see cleavage, you see inflammasome assembly, you see cleavage of Cas1. Again, this very pro-inflammatory caspase that has a lot of downstream effects. Macrophages is very well documented, and that's what we're showing here. However, in hepatocytes, this is using a FLICA dye, which is specific for cleaved Casp1. We actually can induce pretty high levels and clustering of this cleaved Casp1 in these hepatocytes. So they're clearly very active cells in the context. This is in hypoxic context. And when we inhibit that with glycine, we can minimize the clusters of Casp1 that we see produced by these activated inflammasomes. So still kind of ongoing, but kind of exciting in that context that we show that hepatocytes aren't such innocent bystander cells in this process. And so the conclusions from this work relatively earlier that, NINJ1 is a really interesting target for decreasing hepatic ischemia-reperfusion injury. Certainly in an in vivo model system in mice, we can see that it plays a key role. The plasma membrane rupture effect of NINJ1 with this dual function seems to be really the dominant effect in this system. And that if we inhibit NINJ1 pharmacologically with glycine, we're able to recapitulate but not augment that knockout. And that it's hepatocytes as well as macrophages that are potentially playing active roles in this. And so potentially a nice new target in some of the work that's being done. And we have some work now going on in pigs with one of my partners, Dr. Marcus Selsner, as well as some of our human samples, seeing if we see that same phenotype. So that, I think the lab, a lot of this work was done by Bruno Martins, who's upper right-hand corner there, as well as Jan. He's new enough that I don't have a picture of him, I'm sorry, on the, and Sarah Zhao from Ben Steinberg's lab. Sergio Grinstein is one of the senior cell biologists at SickKids, a great research mentor, and thanks to the group, and thanks for having me here today.
That was really fantastic. Thank you very much. I'll just check in virtually. Do we have any questions? Not currently. And questions to start from the floor?
Very, very nice work. I have a question about fibrosis in your NINJ1 knockout model. How does it look like?
Yeah, it's a great question. So we're looking now. So basically, we've been looking at pretty early time points with ischemia-reperfusion injury and looking at this kind of widespread necrosis. So that would have the most clinical translatable effect, in essence, to primary non-function. So when we put a liver in, and we know it's a marginal organ, we put into a pretty sick person because we feel compelled, we got to get this person transplanted. They're very, very sick and their window may be closing. We put that organ in and it just doesn't work. We have a profound kind of this, inflammatory collapse response, and we end up with a process where the INR is, increasing and the bilirubin increasing, and really, this acute severe injury. And so that's probably what this model is delineating the best. We are, though, looking, because we're looking at early time points. Now we started looking now kind of 2-3 weeks down the line. So what's really remarkable, number one, is how much of the necrosis is kind of cleaned up by that point. But there is some suggestion that there is certainly decreased fibrosis in the NINJ1 knockouts we'd expect, because they have a less severe early injury. What we're really interested in is what happens to the innate immune cells in that environment. So there's this concept of trained immunity, and where basically we used to think of memory as being only a component of the T and B cells, but the reality is that a lot of the cells, and macrophages in particular, that make up the adaptive immune system, in particular with epigenetic annotation, they can retain a activated response. And so we're looking by ChIP-seq now to see with the Kupffer cells in particular that we pull out of the liver of those less injured livers, if we see a decreased pro-inflammatory type phenotype. And the reason it matters is we know that with that kind of early introductory slides, when you have a bad early ischemia-reperfusion injury, you're really high risk long-term. Like, those folks are really high risk for secondary fibrosis, acute cellular rejection, chronic cellular rejection. They never fly. We don't really understand why that is. And so some of that may be the priming of that early immune microenvironment.
A very nice talk. So in the original paper, the NINJ1 was described in the myeloid lineage, and you're looking at NINJ1 in the liver. As you know, this lytic form of cell death has probably evolved to prevent certain infections and certain intracellular organisms, viruses as well. So in your case, if you target it, the therapy is not specific to the liver. It's nice that it's expressed in the liver, but there's going to be some in the macrophages. How are you going to target this therapy so you don't impair immunity, especially viruses, which may be relevant in liver transplant?
Yeah, that's a great question. Right now, we're basically doing what we want to do is an inducible knockout, basically. And so what we'd love to do is be able to knock out them differentially in hepatocytes, specifically under the albumin promoter, versus in macrophages and liver under the lysa-M. And so that's what we're working on right now with our TCP, with the big transgenic mouse facility in Toronto associated with U of T, is we'd like to be able to differentiate the effects of the two. We're clearly seeing both, like we see clear lytic cell rupture of both hepatocytes and Kupffer cells. And there's obviously other cell types that we can't see as well because they're more fleeting. Stellate cells, the endothelial cells, we don't, cholangiocytes, we don't see them as well. But for the two predominant cell types with the macrophages, Kupffer cells, we for sure see lytic cell death there. Whether or not one drives and the other follows, or they're both kind of equally contributing, we don't know. And so that's why we want to do a more specific, in essence, myeloid knockout versus, hepatocyte knockout. And so that we can kind of differentiate of those two cell types, which is the more important. Great question.
Do we have any more questions from the virtual? I do have a question, actually.
Please.
How does the clamping of the circulatory, I guess, the circulation in your model differ from transplant situation, if that makes any sense?
I mean, it's doable. I mean, the problem with, transplanting in mice is super challenging. Doing a liver transplant, there's probably only a couple of groups in the world that do this on a regular basis. It's really, really technically challenging, and you probably can't do it without inducing some type of portosystemic shunt beforehand, because they just don't tolerate clamping very well. So it's not easy per se. It's as easy as an in vivo model can be. It's relatively straightforward and it's reproducible. It's certainly different. Like, we're not worrying about, an allogeneic response. We're just basically doing a warm ischemia-reperfusion injury model. And so it has some crossover effect to transplant with that effect, but it's not the same. The porcine stuff that we're interested in doing will be in a liver transplant model. And so this is one of my, two of my collaborators, Marcus Selzner and Nazia Selzner. They do this, as you know, they have a liver transplant model where they basically do, in essence, a DCD liver. They pump it, which is very nice, because then we can look at indicators of liver function on the circuit, and then they implant it. And so we have what glycine is easy, but we're trying to do targeted antibody inhibition of NINJ1 so that we know there's a high level of specificity. And so what we'd like to do is in that context of that model, because it is a limitation to the model. The ischemia-reperfusion injury works well, it's reproducible, but trying to develop a liver transplant model in mice is a whole separate ball of cheese wax. So.
It's really interesting stuff. Given the role of NINJ in other, or the location of it in other places, couldn't you do a heterotopic heart transplant model in mice or lung transplant model like
For sure.
Dan Kreisel's been doing?
For sure.
Has that been looked at?
It hasn't. So, I mean, NINJ is this recent stuff is all relatively new. It was mostly immune infiltration and kind of CNS signaling work. And so we have now within the Ischemia Transplant Center at U of T, we have a animal surgeon who's trying to get up and running, small animal surgeon with heterotopic heart transplant as kind of a service to everybody. So if you have an idea and you want to do that with your animals, that we could do that. It's a great idea because it's a relatively of a vascularized allograft model. It's a really well accepted, like you mentioned. So certainly something that we could do. I don't know how well it's expressed in cardiomyocytes. That's something I haven't looked at, but it seems to be fairly broadly expressed. It just seems within the liver, it's certainly enriched in the hepatocytes and the macrophages, but I don't know in the heart off the top of my head.
That's something CDTRP can help with connecting, because a number of us as investigators use some of those models.
That'd be great.
So those collaborations might be something to consider.
Absolutely. That'd be terrific.
Any more questions? Nothing else, I think. Yep.
Thanks very much.
Check out
Appreciate it.
say it.